Hello, everyone, and welcome to today's webinar, Transformative Insights from your CDD Vault with Cerella's unique AI. So today's presenters are Barry Bunin, CEO of Collaborative Drug Discovery, and Matt Siegel, CEO of Optibrium. So with this introduction, I will now give the floor to Barry and Matt.
Thanks, Abe. Hi, I'm Barry Bunin, and should we go to the next slide there, Matt? Great. So I know CDD Vault's a whole solution for drug discovery data with activity and registration, visualization, inventory, ELN, and Cerella. CDD Vault's differentiated in terms of the ease of use, handling the data, and the secure collaboration, complexity simplified. Cerella is a leading data imputation, AI modeling software. And so maybe, Matt, you can talk a little bit about why would one want to combine CDD Vault and Cerella and use both of them together.
Yeah, thanks, Barry. Be happy to. So we've been working with our friends and colleagues at CDD to make the link between our Cerella platform and CDD Vault, really so that you can take your data in CDD Vault and really derive valuable insights from it using the unique AI approaches within Cerella. So just to explain a little bit about the sort of questions we can answer, they really come down to the challenges of the data that we have in drug discovery that I'm sure you'll all be familiar with. For example, we know it's impossible to measure all of the compounds that we're interested in for a particular drug discovery project in all of the relevant experimental assays. It's just too time-consuming and expensive. So the question is, well, how do I actually make the most from the data that I do have? In data science terms, this is the challenge of data sparsity. Most of the potential data I'd like is actually missing in practice. Equally, we know that there's a lot of variability in the experimental data that we do have. We know that there are experimental errors and artifacts, and we want to avoid being led astray, those false negatives that can lead us to missing opportunities to find high quality drugs, or false positives that mean we spend a lot of time and effort that's wasted pursuing a hypothesis that turns out to be based on incorrect data. And given that we can't run all the experiments we might like to, which are the most valuable experiments to run? What data, if I generate it, will actually give me the most new information on which I can make my decisions? And then also, how can I use that data that I do have to build better predictive models for new compound designs so that we can choose the best ones for synthesis and testing? And so in a nutshell, Barry, this is the way that Cerella can help. Obviously, it's about reducing the time and cost of drug discovery, for example, by proactively highlighting high quality compounds by filling in that sparse data. This is the process known as imputation. It can also increase the confidence we have in the decisions we're making by identifying hidden opportunities due to missing, uncertain, or incorrect data, flagging those outliers and false negatives, translating the insights we can get from these unique AI approaches into planning the experiments to do so that we're focusing on the most valuable measurements to make, and gain more value from the compound data that you do have by making more accurate predictions, particularly of complex downstream and typically expensive experimental endpoints that are intractable with conventional QSAR modeling. So that's what motivated us to really work on this project.
Sounds very intriguing. So how does it all work?
Yeah, okay. Well, let me explain a little bit about the science behind all of this. So first of all, let's start by defining this concept of imputation and contrasting it with the more familiar predictive models that we work with. So predictive models typically start with input features of some sort. So in our case, in chemistry, we typically work with some sort of chemical descriptors that are calculated from the structures of our compounds. So here we have a matrix of all the compounds and the descriptors that we can calculate, and we end up with this sort of orange matrix here, which is completely filled in because I can calculate every descriptor for every compound. In a typical QSAR model, we'll feed that into our favorite modeling method. Here I'm sort of illustrating a random forest, but it could be an SVN, and deep neural network, a Gaussian process, or whatever method you like to use. And then we can make predictions for one or more experimental endpoints for each of those compounds, the sort of purple squares here on the right-hand side. But imputation's quite a different approach. Imputation starts with that sparse data that we do have. So here I've got this matrix of assay data, and the green squares represent the measurements that have actually been made. And most of this matrix is white space. Most of it hasn't yet been measured. And what imputation does is take this sort of limited data that we do have available as input to fill in these blanks, the purple squares here on the right-hand side again. So we're filling in the gaps between these experimental measurements. And the particular method that we use is called Alchemite. This is a result of another collaboration with one of our local partners called Intelligens. And what this does is it still can take the descriptors that we're all familiar with here on the left-hand side. And so it can capture that sort of structure-activity relationship between those structural descriptors and the different experimental measurements that we make, the different assays. But again, it can take this sparse data directly as input. And even though most of that data is actually missing, where it is present, it contains an enormous amount of additional information about the relationships between these different experimental endpoints, these different assays that we run. And what that means is that it can make much, much better use of that sparse and noisy experimental data than conventional QSAR models. So it can fill in these gaps here on the right-hand side, as I described, impute or fill in that missing data more accurately than conventional QSAR models. And as we've explained in some of our publications you see before, we can also train it for what we describe as virtual compounds that have no experimental data because they've not yet been made or tested. It can still take just the descriptors as input and make high quality predictions for those new compounds as well. Another really important point is that Alchemite doesn't just produce a single value for each prediction. It actually generates a probability distribution over potential values. And what this means is it can estimate the uncertainty in each individual prediction. So sometimes you see a prediction like the one on the right-hand side where we've got this very broad probability distribution. And that's the model saying, yeah, I'm not really sure. It's somewhere around here, but it's got a really big error bar on that prediction. Well, in other cases, we see these very sharply peaked probability distributions, like on the left-hand side. And that's the model saying, yeah, I'm really confident in that value. It's got a small error bar. And what we've demonstrated in the publications at the bottom of the page and in many others is a very strong correlation between the model's estimate of its uncertainty and the accuracy observed on independent test sets. What this means is we can really highlight the most accurate predictions on which to base our decisions. This enables us to more confidently target high quality compounds and also to prioritize experimental resources. Because of course, where that uncertainty is the greatest, like here on the right-hand side, that's where extra experimental data will add the most value and add the most information. And finally, it's also really important that we understand the reason behind these predictions. The vogue term now is, of course, explainable AI. You don't want to just get a black box where it spits out a number. So Alchemite actually generates interpretable relationships between experimental endpoints. So here you can see, for example, what we call an importance matrix in the bottom left, where we've got descriptors and assays here that are used as input along the bottom, and the different outputs here along the side. And what you can see is for assay sort of A1 here, the most important inputs are these descriptors. So we can understand those structural features contributing to that prediction. Whereas for assays A2 and A3, the strongest contributor are actually the other assays as well. And so actually there's a strong relationship between those experimental endpoints that tells us a lot of information. For example, it can provide insight into the mode of action. If you have relating a downstream experimental endpoint like a phenotypic result or even in vivo efficacy back to sort of earlier measurements like target activities and so on. And we can drill down for individual assays. So here's for assay A3, we can look at that importance and immediately understand here that this assay A2 is the most important input. And what that means, it also helps us to determine what experiments to run that will add the most information to impute these late stage studies as well. So hopefully that sort of motivates a little bit about how it works. Again, I haven't gone into all the equations, but all the details are in the references you can see at the bottom of the slide here.
Great. So if you share the next slide, I want to discuss why this is an interesting and challenging problem. And these are some slides from Dr. Guy Breitenbucher when he was at UCSF, and Peter Gedek looking at like a free Wilson analysis where you look at the additivity between, say, an R1 and an R2 group. And Peter Gedek had developed this intelligent chemistry browser technology we call SuperSAR in our visualization module of CDD Vault today to look at sibling and cousin compounds instantly by traversing the whole lattice at once. And this was part of our Q3 2019 webinar. And people who make combinatorial libraries think about this type of thing a lot when they generate a full matrix and look at SAR. And so here, for example, in this grid, you can see this R prime at the 2 position is the most potent, and across the board, when you were trying it with just one by one matrix versus the others, this red one would have shown up as the highest if you hadn't filled out, or to use your term, imputed the data. And conversely, here this row here, you looked at the data and this was the best intercept of the 2. But due to non-additivity, you would have a surprise here that you wouldn't have known until you filled out the full matrix. And they didn't really know why this was happening or have any idea about it. It was basically a surprise. You can have steeper SAR or shallower SAR, and then you can have things that are kind of surprises. If you go to the next slide, please, this shows this a little more in terms of the energy values. So here with one R group, this isopropyl, your PIC50 goes up, and here with another variation, in this place, the methylamide has a butyl group off of it and has a similar jump in energy value, binding energy. And one would hypothesize, based on additivity free Wilson, that if it helps, gets more potent here and gets more potent here, you do both of them and it gets even more potent. But in fact, the opposite was observed. You added the 2 together and it got worse. And so they were curious about this, and really interesting. So for this non-additivity, they were able to get crystal structures, and they were able to show that the protein actually moved when one of the R groups was changed, and thus made it harder for the other bulky group to be there. And this, to the best of their knowledge, was the first experimental evidence for non-additivity. And so the fascinating thing about drug discovery, both modeling and experiments, are sort of the surprises or the unknown unknowns. And unless you had data, you could have co-crystal structures, which they had after the fact, and now there's AlphaFold to predict protein structure. But being able to predict protein structure in combination with small molecules, we don't have 100% perfect information today on all of our biological assays and systems and how they interact with molecules, making it still be an experimental science. Now, predictions can work. Certainly a high throughput screen, you can add a model and reduce the number of molecules. And as you mentioned, some of the assays are just too expensive to practically run. So there's a place for models, and there's a place for data. And I guess I wanted to show some of the harder cases to make people think about the scope and limitations of the technology and how to use it most wisely. But back to you, Matt.
Yeah, thanks, Barry. Those are really good points. So I guess there's a couple of things to say. First of all, I've just spotted in the Q&A panel, one of our participants today is Marcus Metz, who points out that he's the a co-author of the paper that you've cited here, so I think it's important we acknowledge that, and I think we can add a proper citation before we circulate the slides afterwards. And secondly, I think the example that you showed previously of this activity cliff, where you get this unexpected jump in activity due to a small modification in the structure, is one of the most challenging examples for prediction. Actually, we published a paper just at the end of last year with International Flavors and Fragrances, showing how this approach of data imputation, where you might just have a little bit of extra experimental data, can really improve our ability to predict these very challenging activity cliffs. So it's really nice to see that example.
Yeah, it's great that we have an author of the paper in the webinar. And yeah, these are just some of the slides from a previous presentation from Guy, but we'll circulate it with the reference. Thanks.
So with that in mind, I guess the next question is sort of, what will the combination of CDD Vault and Cerella actually do in practice? So let me just illustrate that as well. So first of all, Cerella connects directly with CDD Vault, securely, of course, via CDD Vault's very sophisticated API. And what it does is it automatically updates and prepares experimental data for model building. So it applies all of those sort of cleaning business rules and transformations that we need to do to get the best model performance from that raw data. And it populates that sparse matrix. So here again, we've got sort of different experiments across the top and different compounds down the side here. And again, a lot of this is white space because we can't measure all of those different properties for all those compounds. And then having prepared that data automatically, removing all of that sort of time-consuming and tedious effort, it will then automatically build and, of course, update models using this Alchemite method that I described. And as new data becomes available, you can quickly refresh that matrix and update the models so that you're always working with results based on the latest information and remove that burden of sort of manually rebuilding and retesting and updating models all the time. Now, with Alchemite, of course, one of the applications, as I described, is to fill in that matrix, to add those purple squares that I spoke about before. And now we've got a real challenge because that generates a lot of data. We might have many compounds on this side, many experiments across the top. Only, say, 1% of that data is measured in practice. That's fine. But once we fill in that data, we've got what we describe as the massive matrix problem. We've got this big matrix of data, true big data, that we really need to be very easily access and find those interesting results and valuable conclusions that we can draw. So Cerella scales to make it very quickly and easy to retrieve those. And those results are available in different ways. One that I will be using to illustrate this in a moment is via our StarDrop software, which is a complete platform for small molecule design, optimization, and data analysis. But StarDrop itself, Cerella itself has an API, and so you can access the same results, the same data, the same models via any platform that you wish. And so what can you do with it? Well, here are some of the applications that we've really focused on. First is, as I say, to fill in that missing data and very proactively highlight high quality compounds. So, here's a typical thing we might do. We might construct a query for a particular combination of activities and properties. So here I'm looking for activity against my target, good permeability and good solubility. And if I were to go away and search just the experimental data in CDD Vault, I would return some compounds that had been made and tested and meet those criteria. But using Cerella and this idea of imputation, we can find many more opportunities by filling in those blanks. So it goes, aha, I can see you're looking for compounds with these criteria. By the way, here are some other compounds that are predicted with confidence to meet those criteria. So here you can see the white squares are the experimentally measured values. The blue squares here are those that have been automatically filled in with those highly confident imputed values from Cerella. Equally, we can look for these experimental outliers, these potential artifacts. So here you can see in this screenshot, the purple square here is an experimental measurement that has been made, but looks to be highly unlikely based on all of the other evidence in the data within CDD Vault. It says, look, this looks to be very unlikely. It could be an experimental error. On the positive side, it could be some very unexpected SAR, but you might want to look at this in more detail, potentially retest this compound in this assay, because it could really be a winner. You can even drill down. So here you can look at the underlying probability distribution, sort of shown by this blue histogram here. And you can see this compound is highly likely to be active. You know, it's got a nice high peak here to the right, but the experimental measurement is this red line that's way off on the negative tail. So that's a potential false negative, a compound that we might have missed because of an inaccurate or an experimental error. And we can even say, well, maybe there's an assay that is more time consuming and expensive. So here, for example, is a cell-based activity, a phenotypic screen. And I might say, this is my target. I want to predict and choose the best compounds to progress, to really invest my time and effort in only the best compounds. But perhaps there are some other less expensive, higher throughput assays that I could run. Which are the most valuable experimental measurements to make in order to more accurately predict or impute that downstream experimental outcome? And so here you can see it's suggesting, oh, there's a couple of assays. You see these high bars corresponds to the activity against my target and the CACO2 permeability. So if I had to invest in running a couple of assays, those would be the two I would spend my time and effort on. And I can even click here and see which are the specific compounds that it recommends running in that assay to get that coverage of the chemical diversity to more accurately make predictions across the whole data set, and then hence make better decisions about which compounds to progress into that more expensive downstream experiment. And finally, all of the same endpoints are available as predictive models based purely on structure, so similar to QSAR models, so you can use them for virtual screening for new compound libraries or new designs, make predictions across a broad range of endpoints, and then bring that data together, for example, using multi-parameter optimization, so that you're really targeting the best compounds for synthesis and testing. So those are some of the different ways that you can apply this deep learning imputation method using Cerella.
This sounds very intriguing. When it's real, you can actually see it live, and for the audience to see the GUI of CDD Vault with Cerella in action. Is this something that's at a stage where we can demo it and show how it works?
Oh yeah, absolutely. So let me jump out of the slides here and show you a quick example. So this is using Stardrop. This is our software, which of course we've developed a very intuitive, very user-friendly interface, as I hope you'll agree in a moment. And this is connected to Cerella, which in turn is connected to CDD Vault. And we'll talk more about the mechanics behind the scenes a little bit later. But let's look at just a couple examples of the applications that I just described in action. So for example, I'm using a data set here. It's a public domain data set published by MMV called the Pathogen Box. It has lots of different activities for developing world and neglected diseases, and also various ADME data and so on. So it's a nice little playground we can look at. So let's imagine a scenario where I'm working on a project and I'm looking for compounds that are active for tuberculosis. Right. So what I'm just going to do is go back to my project. And I'm just going to perform a query. And I'm going to do something we would typically do. I'm going to look for compounds that are active against in a couple of different TB assays with different experimental conditions, and also retrieve some sort of ADME and physicochemical properties that have been experimentally measured. So I'm looking for compounds that are active in these two assays, and I click OK. It goes away. This is now looking at the data from CDD Vault, and it brings back the results. And so what you can see here is we've got compounds that are active in these two assays here. These two different conditions all have better than 2 micromolar activity, which was the condition that I set. And you'll also see that where it's available, I've returned some additional ADME data as well, but most of that data is missing. So you'll see it's returned 20 compounds here with a bit of data, but it's not the most exciting starting point to find high quality leads or series for this particular project. So far I haven't really used Cerella at all. So let me show you what happens if we do one more thing, which is to repeat that query. Same query as before, but all I'm going to do now is to check this box that actually goes to Cerella and returns the Cerella imputed values as well. And if I click OK, it's going to go away. Takes a couple seconds to do that. And it doesn't bring back 20 compounds, but it brings back 62 compounds. And what you can see is, as well as that experimental data in white, you can see all of these blue squares have been filled in automatically using sort of high quality predictions using the sort of alchemite imputation approach that I described before. So we immediately have a much, much richer data set to explore, looking for high quality compounds with good activity, as well as good physicochemical and ADME properties for our objectives. And you'll see there are some examples here where we've got both of the experimental data points that have been measured, but equally you can see these other examples where we have the compounds we would otherwise have missed, which are imputed to have good activity against these targets as well. So lots of new opportunities. Furthermore, you can see this example right here, this purple square. Now, this compound here was measured to be active in one of these assays, but actually measured to be inactive with a greater than 50 micromolar MIC in this second assay. So this is the experimental result, and normally that would not be returned by a query of our database. But what Cerella is saying is, hey, I know you've measured this to be inactive, but that looks to be very, very unlikely based on all of the other evidence in the database. Maybe you might like to look at it again. And indeed, if I drill down and look at that probability distribution generated by the model in Cerella, what you can see here is that it's sharply peaked down here, well under 1 micromolar, around 0.1 micromolar. So in fact, Cerella thinks that this should have an activity of about 0.25 micromolar with some uncertainty, of course, but it's saying, hey, you might want to go away and retest this because it looks like it could be a very valuable and interesting compound for your project. And here in Stardrop, I can sort of bring all of this data together with all of the uncertainty. And I've got here a multi-parameter scoring profile. This uses our probabilistic scoring approach. So we're looking here for compounds that are active against these two assays, as well as good microsomal stability, good permeability, and good solubility. This is just generating a score for each compound, taking into account the measured and imputed values and all of that experimental uncertainty and predicted uncertainty. And it enables us to very quickly focus in on those compounds with the best chance of achieving a balance across all of these different properties. And again, you can see some where all of these values have been measured, but equally you can see some of these are automatically filled in. We can see they're predicted to be active and have good physicochemical and ADME properties as well. So it really surfaces these valuable new opportunities. And last of all, let me just show you one more quick example, a slight change of pace here, but we'll move over to look at a series of compounds targeting an anti-malarial. This is a series published by the University of Cape Town, but it's an example of how we might use this when we're designing new compounds. So this is a small library that has been designed around a particular series, particular scaffold. And as well as imputing existing compounds with some experimental data, all of these same experimental endpoints are available as predictive models based purely on the structure. So I could, for example, predict activity here against 3 different strains of malaria, both drug sensitive and drug resistant. And equally, you see all the different ADME properties where we've automatically built these models. So I could include, for example, liver microsomal stability, in this case in mouse, permeability, plasma protein binding, solubility as well, for example. Maybe we'll add in some hERG inhibition for good luck. And I can run these models and you'll see it makes predictions for all these different endpoints for all of the compounds. And you'll see these squares here are blue because, of course, we don't have any experimental data here. This is predicted purely based on chemical structure. You'll also notice these white squares. This is actually where Cerella is saying, hey, I can't make a confident prediction. So sort of outside the domain of applicability, it's being completely honest and saying, you know, look, the uncertainty here is too great for you to use that data with confidence. And again, we can bring it together, for example, by scoring these compounds to find those with a good balance of activity and ADME properties. We can sort this. We can start to look for the best compounds that are the compounds that are predicted to be best for synthesis and testing. And where we see shortcomings here, for example, you can see this blue bar here is low, indicating a potential issue. It's an activity against a drug-resistant strain of malaria. We can start to say, well, can I actually redesign this compound, improve its properties? And so this works in a very interactive way, just to show you the models respond very, very quickly. Here's that compound I've just selected, and I can start to make a change here, for example. I could change this into, for example, an imidazole very quickly, just by editing away. And you'll notice every time I click, the predictions update in real time. And I can just draw that in there quickly as I can to save a little bit of time. And there you can see here, the overall score now has improved from about 0.21 to about 0.25, and the activities are getting a little bit better, and some of the ADME properties are at least as good, if not better than they were before. So you can still use this in an interactive way, connect it with generative models, for example, and use that to guide the automatic generation of new compounds, for example, in our Inspira software. So again, really, really quickly, but hopefully you can see how this enables you to look at data in a new way and surface some valuable and interesting results in a really intuitive way.
That's a wonderful demo. Thanks for sharing. Do you have any information on how it works with commercial laboratories and experimentally as well?
Yeah, that's a really good point. It's all very well, looking at all of the clever software, but does it work in practice is an important question to answer. So just to mention, we actually ran one of our own webinar series, I guess it was about a month ago, where we were talking about applications of this approach, throughout that sort of discovery process from early hit all the way through to sort of preclinical development. I'm sort of showing the timeline here with the examples that we actually illustrated in that webinar, and some of our collaborators with which we've worked on this. But let me just give you a couple of little snapshots of these, a few of these, just to illustrate the sorts of results that we've seen. And on these slides as well, I have references so you can dig into more detail and links to more detailed webinars. So one is an example here where we were working with Constellation Pharmaceuticals, and we looked at an application really focused on sort of optimization projects. Actually, we had a data set of very heterogeneous data with target and phenotypic activities, ADME endpoint as well for two projects. It was quite small. In total, by the end of these two projects, there were about 2,500 compounds across these 18 different experimental endpoints, but it actually started with a much smaller data set. And the first thing we did is we compared the performance of this method, Alchemite that's in Cerella, against different QSAR methods, and showed even when we were looking for the best QSAR model, using lots of different methods for each of the different individual endpoints, a really substantial improvement in the accuracy by using this imputation approach. So the average coefficient of determination increased from 0.5 to 0.72. But the really big improvements we saw, for example, were in these much more complex and more expensive endpoints. For example, here's a cell-based screen, a phenotypic activity. Here you can see the best QSAR method. We tried lots of different methods. The best one on the independent test set actually had a negative coefficient of determination. What that means is that the predictions are worse than generating random numbers. So really not very helpful at all. But we can understand why. A phenotypic activity is the combination of one or more different target activities plus perhaps cell permeability, protein binding, solubility. Lots of different factors contribute to determining that phenotypic activity. And it's just too complicated for a conventional QSAR model to understand and really dissect that structure-activity relationship. On the other hand, Alchemite achieved a really very respectable result. I think the R-squared was about 0.67 on the independent test set. So really a very useful model. And the reason is, of course, it's learning about these relationships. Even though the data was very sparse, it was understanding the relationships between those different assays and hence able to make a much more accurate prediction. We published this in Journal of Chemical Information and Modeling. We also gave a webinar that went into much more detail about the different ways we explored these data and the applications of the method. But we were joined in that by Julian Lavelle, who was their vice president for drug discovery, who explained how they actually used the method and the value that they saw. And one of the things he said was that actually, if they'd focused on the compounds that were confidently predicted to be inactive, they actually found almost no false negatives there, and they could have saved themselves, I think he said 12 FTEs over 3 months in unnecessary synthesis time, which conservatively is about $600,000 in time and effort they could have saved. Here's another example close to our heart, working with the University of Dundee, looking actually at global health diseases. In this particular case, we were looking at an anti-malarial project. And what we were doing here was working with a data set of 300,000 compounds, so much, much bigger, and 500 experimental endpoints across a broad range of different diseases, and also focusing particularly on a project, an anti-malarial project, anti-TB project again. Here we were looking at just much smaller data set within that global data set of just about 500 compounds and 34 endpoints. And one thing we wanted to see is, well, how well does this global model perform when making predictions just for that project? One of the challenges we see in modeling often is that if we build a model across a very large, very diverse data set, it doesn't apply very well at resolving within an individual project. So we compared that global model with one based purely on this project data set and saw that there was no significant change in performance for imputation. And for the virtual model, we actually saw an improvement in performance when using the global data set for making predictions within the project. And so this means we can literally build a single model of this very large data set and run it on all of these individual projects, sort of build once and run everywhere. Another example that I'm showing here is the ability to rely on the confidence in the predictions, the error bars produced by the models. And so what you can see here, for example, is for an independent test set looking at an MIC endpoint. Here's the correlation between the experimentally observed value and the predicted value. So you can see a nice correlation here. The orange line is the line of perfect prediction. And you can see here the scatter is very tight around that line, and the error bars for each individual prediction. And here at the top, I'm showing a graph where here for 100% of the data set, this is what we're looking at. You can see the accuracy here, which is the root mean square error of about 0.3 log units. But as we move from right to left along this axis, what we'll do is focus on the predictions that the model has the greatest confidence in, the smallest error bars. And what you can see here with this blue line is that the accuracy increases, that the root mean square error goes down. And so if, for example, we move to look at the 50% most confident predictions, what you see is they snap in even tighter to that identity line. So again, here's the 100% and then 50%. And in fact, if we go even further, we can look at the 10% most confident prediction, and almost all of these lie right on that identity line. So really, really accurate predictions. But the eagle-eyed amongst you will notice that we have this tick up at the left where the accuracy actually gets worse. And that corresponds to these two data points here that I'm circling. And those are those outliers. They have a really small error bar, and all the other points with the small error bar are very, very close to the observed value. So these are ones where it's really worth going and looking at experimental data, because it could be a really valuable compound with greater activity than was measured experimentally. And last of all, we've also looked at applications, for example, with generative methods for exploring new optimization strategies. Another anti-malarial activity example with the open source malaria project here. And what we did is we built models of a very sparse anti-malarial activity data set here. We then use that to generate new compounds, actually using what we call the NOVA module in Stardrop to explore new compounds around a particular chemical series, sort of expanding out around it. This was actually run as a blind competition. We were able, along with other organizations, to submit compounds for synthesis and testing by the open source malaria project. And what we did here is we did not submit the compound predicted to be most active. Instead, we submitted the compound most confidently predicted to be active, using that ability of the model to understand where that confidence is high enough. And indeed, what you can see here is of all of the compounds submitted by the different organizations, the only one experimentally confirmed to be active was the one that we proposed. Again, this has been published actually most recently in JMed Chem at the end of last year. We did a webinar where we were joined by Professor Matt Todd, who leads the open source malaria project. And he said something really interesting, is that this compound that we propose is one that he didn't think any of the humans involved in the SAR of this project over many years would have bothered making. The particular suggestion that we made here was actually substituting this tertiary butyl group. And he said that he and all of his colleagues thought that was going to be inactive when they saw it. And actually it gave them a new route for optimization across this project. So those are just a few quick examples of the application. And there are many more. If you go look at that webinar we ran a few weeks ago, we go into about 7 or 8 different examples.
I think there's still one more example.
I apologize.
With,
I was going to summarize
on it, and
yeah, you're absolutely right, Barry. Apologies. Yeah, I was just going to sort of summarize. These are the sort of different applications that I spoke about earlier, and just again, for references, we'll make the slides available later so that you can sort of go and find the original publications and the webinars that describe this in more detail. Thanks for the reminder, Barry.
That's great. That is just an impressive list of examples and really shows the breadth and depth of the technology. So I'd like to segue to how one would link CDD Vault and Cerella. Maybe, share a little bit on the CDD Vault side. So here you can see CDD Vault's a whole solution for drug discovery data management, and a lot of groups use it as their whole solution, and others use it for secure collaboration. You can see on the bottom left the image with the Bunsen burner and the dose response curves. So the electronic lab notebook handles chemical reactions. That's sort of the Bunsen burner round bottom flask image, including stoichiometry table and the protocols, if you will, the metadata around the data. We have a high profile fair data project with the Pistoia Alliance, where we annotated a number of PubChem assays, provided to the PubChem classification tree. So it's also an electronic lab notebook for biologists and biological protocols. The activity and registration modules, really the main database in CDD Vault, and the activity has sort of a double meaning of capturing the activity from the biological experiments, such as the microtiter plate data, ZZ prime statistics, IC50 curves, hill slope, etc. But also the activity of being a collaborative vault that medicinal chemists and biologists experimentalists, computational scientists can all work together. And the core registration capability works if you want to generate your registration numbers with CDD Vault or mirror another registration system. And the forklift is the inventory for tracking when to get additional samples, either milliliters, microliters of in DMSO of samples, or fresh molecules. And then the visualization, the last icon there, has the scatter plots and histograms and bar graphs. It also has the SuperSAR, the technology mentioned for looking into additivity and non-additivity. And this has become very widely used platform. This is the first hosted drug discovery data system before AWS existed. I used to joke around that we got all the arrows in the back of no one trusting hosted cloud system, and now with everyone moving to it, copying this. In any case, now with over 3 million logins from paying customers, it's become very robust, reliable system for a lot of laboratories around the world and every major continent. If you can go to the next slide, please. And we expanded from small molecules and the SAR around those to biologics. And the exciting thing is the same 4 modules, ELN, activity and reg, inventory, visualization, are the same, whether you're screening a protein or a small molecule or various other types of therapies. And so we announced integration with the SnapGene server for DNA files. And so now researchers can have a small molecule CDD Vault, a biologicals vault. We also came up with this Minchi and mix file. We worked with the nonprofit to come up with a computer readable format for looking at mixtures. If you want to look at screening combinations in a computer readable format, formulations, consumer goods, etc. So the Vault is now versatile, whether you're using it for looking at your biologicals or your small molecules. We really focused on making it easy for project teams to look at the data. And that's why we're so excited to partner with collaborative drug discovery. We'll collaborate with all the companies that provide different mining and modeling technologies, as you saw in the webinar today. And so I guess a big question for researchers is just how can they try this themselves with their own data and their own ability to create models.
Yeah, absolutely. And just before I do that, let me just show you what's going on a little bit behind the scenes as well, because we've been delighted to work with our colleagues at CDD to really make that sort of seamless link between CDD Vault and Cerella, so that you can get your data into Cerella and sort of unleash all of these new methods very, very easily. So let me just give you a quick insight into what's going on, so you can see how easy that process is. So I'm just going to log into my CDD Vault here, something that I'm sure many of you in the audience will be very familiar with, and just to show you the saved search, where I can just show you that pathogen box data that I was just showing you is all here in the Vault, all of the different compounds, all of the different experimental endpoints that we've just been discussing. And of course, a lot of that data is very sparse, as we've seen. A lot of it's missing, but it's all there within the Vault. So let me head over to Cerella. I'll log in here as well. And this is the sort of the administration interface and also the data scientist interface. If I want to add a new data source, I can click here on the data sources. You'll see I've actually got the pathogen box data in 2 different sources here. One's a data file, the other is directly from CDD. But just to show you the process I went through, if I want to add a data source, I can add a data source from the Vault. So I'll call it pathogen box CDD2 because I already have a first one. And then I'm just going to enter the API token here for my Vault, set that token. I can say it's the Optibrium sandbox, and then I can connect. And that will just take a little while. So what you can see here is a little progress bar. That's going to take a few minutes to bring down all of that data into, it's 10% done already. But just to save us a little bit of time, here's the one that I did a little bit earlier. So I'm just going to click on that. What you'll see is it's brought in all of the structures, all of the data here. And for each of these endpoints, you can see where there is data. But equally, you'll see it's automatically brought in all of that sort of metadata, like the measurements, the units, and you can go through here if you want and set some additional information for Cerella to use, but it pre-populates that all for you. It's already in there, ready to go. And if I wanted to then build a model, all I need to do is go here to systems data and click start. It will ingest all that data, do the hyperparameter optimization, the training, the prediction, the testing against the independent test set. And then you can drill in here to see all those sort of validation results that you might want to look at, for example, these sort of rollback plots that I was showing you before for a particular sort of any endpoint that was of interest to you. Yeah, so for example, these sort of plots that I was showing you earlier. So again, this is for the data scientist who wants to make sure everything's working as it should, but it's a really very, very quick and very straightforward process. And then you can access all the same results that I was just illustrating in my live demo. And of course, as you say, Barry, if anyone in the audience wants to try this, we're very happy to work with you to evaluate Cerella with your own data. We'll set up a secure instance for you to use this. We can help you to establish the link in the way that I just showed you. We'll help to monitor the building and validation process, provide you with training, and then you can have a couple of months to try it yourself with your own data via Stardrop, and we can provide complimentary licenses for that. We do actually have a demo server, the one I was using for my demo, which you're welcome to try if you prefer. It's already pre-populated with that data. And if you're interested in that, you can contact us at info@pibroom.com. If you're not already a CDD Vault user, then of course the contact there is info@collaborativedrug.com. So hopefully that was useful and interesting to you. I think, Barry, we're on to Q&A.
Yeah, thanks, Matt. That was an excellent overview. And I'm really glad you were able to show all the technology live so people could see it for themselves. One question that comes to mind is, how much data does one need in CDD Vault to use Cerella for modeling? And you showed examples, both global and project specific. And so, yeah, I'm actually really curious.
Yeah, it's a great question. It's one of those questions where the answer is it depends. But let me just give you some parameters on which it depends and some sort of rules of thumb. So first of all, typically, because imputation can leverage that additional information about the relationships between endpoints, it does require typically less data to get a useful result than a conventional QSAR model. But it does depend on the complexity of those relationships. So if there are strong correlations in there and strong signals, then certainly you can get away with tens of data points for each different endpoint. And of course, they don't have to be complete. They can be sparse, as we've just discussed. I guess we're typically more comfortable in the sort of low hundreds, but that's certainly a very reasonable range of values to work with. The absolute lower limit, Cerella will refuse to build a model of an endpoint with less than 20 data points. The simple reason is it does a training test set split, as it must, to validate the models. It takes 20% in a test set. And of course, 20% of 20 compounds or 20 data points is just 4 data points. And that's the absolute minimum to get any degree of understanding of the correlation there. So that's where we absolutely draw the limit. We're more comfortable once you're up above about 50. Makes
sense. Well, thanks for explaining also how it works, so people have some sort of intuitive feel for the numbers. One of the questions from John Clucker here has to do, people always talk about Lipinski versus non-Lipinski compounds. Maybe you have a disease in the gut. And he was asking, can you speak to any examples of Cerella handling a data set of non-rule of 5 compounds, or if you've looked at that at all?
Yes, I can. I have to be careful here because these are not examples that we've been given permission to talk about publicly. So I'll dance around this a little bit, but I'll give you a flavor. So we've done a couple of projects looking at both linear and macrocyclic peptides. One in particular, we were helping that project to look for compounds with a better PK profile. So they had really good activity against their target in this particular case, but they weren't able to achieve the PK that they were looking for in their animal models. What we were able to do is build models in the way that I've just described based on actually for the PK historic data across a number of different projects, but also bringing in that project specific data. And then make predictions for the activities, ADME properties, and pharmacokinetic properties that they've measured during the course of that project. I think we screened about 100,000 virtual compounds that they enumerated around the particular series of interest. Based on those, they chose, I think, it was about a dozen compounds for synthesis and testing, which they took all the way through to in vivo pharmacokinetics. And I think it was 8 out of those 12 compounds actually achieved substantially better pharmacokinetic profiles, and actually several of them met their sort of their selection criteria for a candidate. So they were really able to move that forward in a very much more effective way.
Yeah, we're at the top of the hour here. I guess last question. You have that nice image at the bottom with the colorful purple and blue squares with CDD Vault, StarDrop, other platforms, Cerella. Maybe you can talk about how one will compare the predictions to actuals. What are sort of the workflows that you see in real life here?
Oh yeah, absolutely. So one of the things that you can do very easily in the sort of administrator interface, it automatically does both internal validation and an independent test. So you can very, very quickly and easily bring back all of that sort of independent test data for all of the different endpoints. So you can just bring back for the imputation model, the virtual model, download that CSV and do all of the analysis that you might wish for independent validation. Obviously, within StarDrop, we make that even easier. So here, for example, is the data set we were looking at before. So let me go back to this imputed data set. If I were interested, for example, in looking at that for, say, one of these activity endpoints, let me just reset the color scale here so it's a little bit clearer. What I can do is actually do what we call a Cerella scatter. And then what you can see immediately is the data points. Here's the experimentally observed values. Here are the predicted values. And then you can see the error bars on those as well. You can see these here are the qualified ones that are predicted, I think, greater than 50. They're not predicted to be completely inactive, but you'll also see these have the biggest error bar. That's where the model's saying, I'm not really sure about this, be careful. But you can see a very, very good correlation within the active compounds in this set of compounds. So yeah, you can very easily drill down into these different endpoints sort of very, very easily and generate these sort of validation plots.
Well, that's excellent, and I appreciate that you're using these neglected disease, TB, and malaria examples. We've been working on humanitarian as well as commercial drug discovery with leading researchers for over a decade ourselves, and I think it's important for the scientific community to keep both in mind. I don't have any further questions from the people attending the webinar. So unless there's any other questions, Abe or Matt, I think this was a wonderful example of the depth and breadth of the two technologies working together. So thank you.
Yeah, fantastic. Well, thank you very much, Barry and Abe and everyone in the audience. It's been a pleasure to speak with you today.